We've covered a lot of ground today, and each one of the talks have sort of addressed really critical points in this very multifaceted um, problem. So I wanted to start with just a little perspective before telling you a little bit about our work, and then trying to weave in the discussion today into the work that we do and some thoughts and ideas. So we are talking about numbers that are so big that it's really hard to wrap your head around the amount of global emissions, the rate of it on a yearly basis, and only increasing. And it suffices to say, I guess, the energy sector and the transportation fuel remain a pretty prominent portion of that. So, any alternatives we can provide for that sector will be very meaningful. In the US, petroleum is the primary source for transportation fuels and chemicals. And if you look at it from the point of view of a barrel of gas, a dominant portion is gasoline. And for that, there are actually really promising alternatives. Tesla came up earlier in conversation. But there are parts of this barrel that could really benefit from some biomanufacturing efforts, jet fuels, and some products, not only for reducing greenhouse gas emissions, but overall, as someone pointed out earlier, to offset many other poor practices for our environment. So, with that perspective, just looking at some numbers, what do those numbers kind of look like? Do they even begin to make a dent in the kind of emissions we are trying to reduce? Here are some numbers. It's from a report from a little while ago, but I think these numbers still hold. 1 billion dry tons of sustainable biomass can make 50 billion gallons, which is about 25% of the transportation fuels a few years ago, and can reduce CO2 emissions by about 450 million tons. And there are other benefits in offsetting chemicals and creating jobs, all of which are sort of part of this socioeconomic scientific engineering challenge. So, with these in mind, the Department of Energy funds 4 bioenergy research centers, and there's been considerable investment on their part since 2007. And we are basic science research centers, and our mission is to really develop the core science technology knowledge bases that are necessary for all of these different pieces to convert sustainable lignocellulosic biomass into fuels and bioproducts. So it's a basic science institute. So what do we really provide? We're trying to figure out how can you make non-food bioenergy crops that have certain attributes. You're trying to come up with deconstruction methods so that you can depolymerize this renewable carbon source into intermediates that can be used downstream, whether by microbes or Kasim's really nice in vitro methods, engineered organisms that can use a multitude of carbon sources with sort of an eye on the final price that one might be able to deliver biofuels at costs that are economically viable and processes that are scalable. But what we are really providing, in my opinion, is options. If you have many, many different kinds of energy crops with many different attributes that are suitable for different geographies, if you have different ways of deconstructing them, different methods to make different compounds and fuels, then you can mix and match them in configuration that matches your application or where you are in the planet, because this is not a one size fit all problem. You really need to have these options. So, what's an example of such a configuration? Here is some work from JBay where we compared wild type and engineered plants that are engineered to accumulate not the sugars, actually the aromatics, and then we use deconstruction methods that can create pure streams of carbon sources, both sugars and aromatics. And sort of the key piece in this is the selection of an organism that can both be engineered for downstream advanced fuel production and can consume any configuration of your carbon source, mixed carbon source, just the sugars, just the aromatics, all of them. So these workflows give you a sort of like a proof of concept or a prototype that shows that this kind of workflow is now possible. So, the reason I point out that particular example is because it sort of exemplifies two things. One is that plant-based biomass from non-food sources can vary considerably and require very different deconstruction. The method that you use to deconstruct poplar is going to be a little different from what you use and get from, say, sugarcane. So you really need options. You need to have a large number of engineered processes, both upstream and downstream, to use the most abundant, sustainable, renewable feedstock available to you, not just the one that everybody worked on for a long time. You need a lot of different solutions. Secondly, lignocellulosic biomass is mainly sugars and aromatics. So we really, usually just think about sugar conversion, and the aromatic parts are usually neglected. But when you're trying to address this massive problem, the kind of numbers of carbon emissions we are looking at and the fuel use in the country, you really can't leave anything on the table. You really have to efficiently convert everything. So, furthermore, um, lignocellulosic biomass is in the most abundant carbon source. You have gas feedstocks, ag waste, municipal solid waste, other waste streams. Really need options to convert all of these materials into final products if you want to make a dent in this problem. The other thing is the consideration of the microbe. Already I said, hey, your microbe needs to be able to use all of these components and tolerate any inhibitors that come along with it, not just the sugar stuff. And it needs to be engineerable. But there are other considerations. There are certainly biomanufacturing targets that could really offset some of these greenhouse gases. And can I make any product in any organism or using any process? Not really the case. There too, you have to kind of match make a little bit. And I wanted to give you just one quick example. This is a bioproduct study. These are some numbers I pulled from the sustainable garment company that is trying to offset all the effects of just a single pair of jeans. And these numbers are theirs, not mine. But really, if every jean is putting 13 kilos of CO2 into the environment, that is probably something worth looking at. The garment industry flies under the radar, but is one of the biggest contributors in industry towards both carbon emissions and environmental pollution. So a lot of people are trying to target the chemicals around the garment industry, and we picked this one chemical called indigoidine to make. Now, is this pigment really the best candidate to reduce greenhouse gas emissions? I really don't know. We need to know a lot more about compounds like this, how they will be separated, how they will be deployed. You need techno-economic and life cycle analysis before you say this is the one or these are the ones. So that's an important consideration. But the thing I wanted to point out is that one of the most common industrial hosts, Saccharomyces cerevisiae, would have been a terrible host for this particular compound. In fact, any other host we picked would have been better than Saccharomyces because of the metabolic sort of underpinnings of these organisms. And that is another thing we need to take into consideration. So we actually engineered that same nice red yeast that I showed you earlier to make this compound, and it made a lot of it, and we were very happy. But next came the problem that Tim talked about. Is this going to scale up? What happens when I put it in a massive bioreactor? Is it just going to uh, extinguish and do something else? So we really wanted to engineer it to make sure that it could scale. But we don't have that many engineering tools in this very new oleaginous yeast. So instead, we turn to an organism where we do have engineering tools and still had those attributes of being able to eat lots of different carbon sources and so on. And we were able to engineer it. And I'll be happy to tell you more about what we did. But the nice thing we could do is show that this particular strain could not only produce more, it produced more sooner, and it could maintain these attributes across various scales. So, really happy with that. And the thing to remember here is that coming to this point in the study required microbiologists, computational biologists, molecular biologists, geneticists, bioreactor specialists. It was big team science. A lot of people came together just to get this little academic paper out. And these were our long term goals. It's taken us a while to come to this point. And we always need partners. We always need partners to demonstrate that our product or project delivered something useful. And sometimes those partners are lacking. Thankfully for us, we have another DOE funded user facility that we use downstairs, the ABPDU. So I'd like to tell you a little bit more about these other programs and projects that we leverage. One is the ABPDU, and the other is the Agile Biofoundry. The Agile Biofoundry has the goal of making the process of strain development really fast. Again, what Tim talked about earlier, it takes a long time to develop these strains, so you have to wait a long time, and so they want to reduce this time by taking data-driven approaches to improve strain engineering, and they want to address a large range of products to improve this process and also increase U.S. industrial competitiveness, and understand that a lot of the past learnings don't actually transfer well, and you do have to do these processes afresh. And the ABPDU has been our partner for quite some time. They routinely work with companies to kind of de-risk their processes. They have found enormous support both from helping industry raise non-dilutive funds and securing their own private financing. I have to tell you, the ABPDU has been instrumental in us kind of getting sort of like past the laugh test in many of our work, and I think there is a really sore need to amplify units like the ABPDU, and Deepti Tanjore, who leads this facilities in the audience today, and she can tell you more if you're interested. So I just wanted to give you these few examples along, why it's important to match the organism with what you're doing, why you need to really know what your most abundant feedstock is, not just what you want to work with, and the issues around scale up that need to be addressed using partnerships. And sort of in my point of view, all of these barriers that we addressed earlier in our discussions with Henry and David are actually interrelated. You can't have one without the other. If one of these sort of bullets are not addressed, you're kind of not being able to address the other points. They're all interrelated points. So I really wanted to wrap up with a couple of next steps. I think overall, we all agree there are no silver bullets. You have to do what you can do in that industry and in that arena. Renewable jet fuels are certainly going to be a key piece in greenhouse gas emissions, reducing them. But I think a careful selection of non-fuel targets can be very helpful. We do need to consider the most abundant carbon sources, not just the ones we want to use, and we have to figure out the hosts and processes that match that feedstock. And we have to start with the scalability and the separations of the product in mind, because at the end of the day, those are the Achilles heel of the process. I think Liz pointed this out earlier, that federal funds do support all of these efforts, but in separate pockets. And we really need cross-interagency work to bring these pieces together. And for successful tech transfer, to get the industry to adopt our process, some pieces need to come together. These are interdisciplinary team science, and the end-to-end optimization take a long time. And I feel that the funding becomes shaky just as you are becoming sure of what you are doing. So, this long-term vision and long-term funding and scoping is absolutely necessary. Otherwise, we get stuck in this rut of short-term deliverables. And also long-term science require a different set of incentives and metrics to measure success. There are a lot of academics in this room, and I think they appreciate what I'm saying. If you get stuck in the rut of putting out the next nice paper, then you cannot address these long-term goals, not to the level that we need to. And then finally, workshops like this are very important because they bring people of different disciplines together. But like I said, some of these problems are socioeconomic problems. So I think we need some social scientists in this group. So that was sort of my last thought, and I'll unshare my screen.